looking at the worksheet you did yesterday, your first protein should be methanone. I can never pronounce that correctly. Your next one should be tryptophan. Then you've got asparagine, which always makes me think of asparagus, valine, isoleucine, and then you have a stop codon. So did you all get the same things? I think that's pretty easy, but you should know how to translate codons into amino acids. There have been questions on the test like that. Now, the question that they ask in number 1, what if you replaced the base in the original position? What if it mutates to an A? How would the sequence be affected? And I'll show you kind of what I did to show that. If this, in copying, somehow mutated, and you had an A there instead of a C, that would change your mRNA codon, and then it would change your transfer RNA codon, and it changes the protein, and you get cysteine instead of tryptophan. So, I actually wrote down here cysteine would be placed in the protein in place of tryptophan. Now, who knows how that changes the protein? That might change the protein in a way that the protein doesn't do what it's supposed to. And that could be a change for the better, it could be a change for the worse. Maybe that's a change that makes blue eyes over brown eyes. Or maybe that's a change that doesn't allow hemoglobin in your blood cells to form correctly to deliver oxygen to your cells. That's a bad change. Okay, the next question. Suppose the base in position 2 got shifted to 16. How would the sequence be affected? So for myself, I drew a little, here's position 2. I drew a little arrow for myself. Now that base is already an A. So it depends. If nothing replaced this base, then you suddenly no longer have a start codon. If this is now TCA, because this got moved, you don't have a start codon anymore. Maybe no protein was ever formed. That would be bad. That would be not a good thing. If the base in position 6 changed to a T, how would that affect? Well, that would actually make a stop codon. If this was a T, then you have UGA here, and that's one of your stop codons. So you would have a protein that was missing most of its length. You would have this little 2 amino acid protein. And again, that different protein could be a great thing, it could be a terrible thing. Who knows? Who knows? Okay, and for number 4, you're just matching the mRNA. Number 5 is a little weird because the sequence is over here. So for number 5, how many codons are there in that sequence? 4. Yeah. So that's going to be a 4 amino acid protein. And then you can go look up the amino acids yourself. If a gene contains 15,000 nucleotides, those are bases, so it's 15,000 letters long. What's the maximum number of amino acids that could be coded for? 500, yeah, because it's 1,500 divided by 3. If another gene has 3,000 bases, what's the maximum number of amino acids that could be coded for?
1,000.
1,000, right. Because every amino acid requires 3 bases. Okay, I will let you do the questions on the bottom on your own. I'll give you a minute to finish those up. And then we're going to start into talking about some basics for cells. So prokaryotes are bacteria and some other things that are like bacteria. So and bacteria-like organisms. You should be taking notes on this. So prokaryotes are bacteria and bacteria-like organisms. And you know what their cells are like? Stupidly simple. They have basically three things. Well, four if we count cytoplasm. So they have a membrane. There's something to keep stuff in the cell. They have a single strand of DNA. Basically, they have one chromosome. And it's not even really a chromosome the way we think of it because it doesn't have that protein understructure. It's really just like a loop of DNA. And they've got a few ribosomes to build more parts to the cell, to build cell membrane, etc. That's it. There's really not much there. Can you believe that something that simple can make you sick? Something that simple can kill you. It's amazing. So there are a couple other things that are what are called archaebacteria, which aren't technically bacteria, but they sure look an awful lot like bacteria to the untrained eye. So those are prokaryotes, and they don't have a whole lot of stuff. Then there are eukaryotic cells. This includes plants, animals, fungus, and protozoans. So you all know what a plant is, right? You all know what an animal is. You all know what a fungus is. Name me a fungus. Mushrooms. It's morel season. They're a fungus. Mold that grows on your bread in the refrigerator. I just had some moldy cheese I had to trim up last night. Those are all fungus. Mildew is a fungus. What the heck is a protozoan? Single-celled organisms like amoeba and euglena. They're little things that are single-celled, but they're still complicated single-celled organisms. Okay, so all of these things are eukaryotes. And their cells have some things in common. Now, in general, we see plant and animal cells represented a little bit differently. Let's see, I'm going to do this. And when I say animal, name me an animal. Deer. Name me another animal. Okay, did you say deer again? Oh, beavers. Okay, name me another one. Humans. Name me another one. Cat. Name me another one. Dog. Boy, nobody's hitting the most common animals on this planet. Huh? Insects, actually. Insects are the most common animals on the planet, most abundant. So, butterflies, ladybugs, cockroaches, fireflies, springtails, little tiny soil insects. Insects and invertebrates are the most common things on the planet. So animals include all of those things, and an animal cell and a plant cell both have a membrane. Okay. In addition to a membrane, plant cells have a wall that gives it structure. They have one really big sort of new development in the world of cells, It's where you keep all the DNA. What is it? Nucleus. So eukaryotic cells have a nucleus. And that's one of the big ways to tell them apart from a bacterial cell. Bacteria have no cell nucleus. They don't really have much in the way of organelles. Sorry, my computer's in the way. But eukaryotes have a nucleus. That's where they store their DNA. And they've got a whole bunch of other organs. So plants, for instance, have chloroplasts, where they photosynthesize. Some protozoa have chloroplasts too. Anything that performs photosynthesis has a chloroplast. Both of these cell types have ribosomes, little scattery bits. And they have all kinds of other things. They have, do you remember any of the cell organelles from when you did this in 6th grade? Remember ER? What's ER? Endoplasmic reticulum. Golgi apparatus, vacuoles, all that stuff. Not so worried that you'd be familiar with all the specifics of that. We don't have time for that, frankly. There's one more that we have not mentioned. What makes power in a cell? Mitochondria. So in a non-photosynthesizing cell, mitochondria are the power plants. And in a photosynthesizing cell, of course, it's the chloroplasts. Mitochondria. Okay. That's about where we're going to stop with this. We're going to go on to a practice question together.